Hello, welcome to Talking Simulator. I'm Cameron, and I have a cup of coffee and 2 cookies because I've been good today. Well, that's not really true, but we're here to finish, maybe, playing in other waters, or I am. Corey is away today, so I am flying solo. How are y'all doing? Which mouse? So, in other waters, where do I want to begin talking about this game? Because it's good. I really like it. It feels a bit like an old text adventure, to be honest. But that's selling it quite short because its interface is really sophisticated and an important part of the way it tells its story. So here we are, the dive AI for a underwater pressure suit that is in the employ of Dr. Ellery Vass as she explores Gliesa 667cc in search of her friend, former lover, colleague, Minay, who we have found. We have found her. Here she is. There she is, all over here. And we have discovered on Gliesa 667cc extraterrestrial life, which has occurred nowhere else in human explored space. The galaxy, as far as we know it, is lifeless except for Earth, which is dying, and now this planet. Interestingly, we thought that Minay may have been the first person to explore this planet, but in exploring it ourselves, we have found that Minay was not the first person here. In fact, there was an entire Baikal exoplanet extraction arcology here that was in ruins. So what happened here? What happened here? And what do we do now? Oh, coffee is just such a wonderful thing to have. Yes? Hello, telephone. I'm sorry. And like, the richness of this world is, well, it makes me very happy, at least. I don't know about you, but the fact that we are literally exploring it and helping Dr. Vass to catalog its life, and then watching her develop theories of what the interconnectedness of this biosphere looks like, makes me very happy. And can I just say once again that I love how the interface makes this world not threatening. I mean, this game, for whatever reason, seems to get a lot of comparisons to, oh, what's it called? The other underwater game where you're on an alien planet. But it has a completely different Subnautica. People are like, oh, this is just a Subnautica clone, and it's like, have you played either game? So we have other objectives to try to get done here. We weren't able to completely examine the arcology last time. But Subnautica, like, gets a lot of mileage out of making its world threatening and frightening. And this interface kind of occludes that for us. Especially since Dr. Vass isn't terribly scared by things either. She's a professional. She's not afraid of this world. And we don't perceive it in a way that really lends itself well to terror. Although maybe that will change. Anyway, let's get to it. And here we go. Maybe we can explore somewhat more efficiently this time. But we can't call for extraction here. Our drone won't be able to reach us. We have to be able to get outside. Oh, all right. It's starting to drain us slowly. This environment is drains our oxygen supply. So let's ascend here and see what we can find. And I'm not saying that this is entirely without menace. I'm saying that the interface occludes that for us a little bit and lets us behave in a different way than we would if we were, I think, perceiving this with our meat people eyes. I thought we had been able to explore this fairly thoroughly, but I think we were getting lost. This was where we came up. There's someplace new. The entire ceiling of this lab has come down, filling the chamber with sand and rock. No one could have escaped. Rusted shut, these hulking metal containers must have carried engineering suits or fabricators. Baikal was building something. I did tweet. Perhaps you can see our tweet if you go to twitter.com/loadingreadyrun. This huge entryway suggests that this is the complex's main lab. Fated Baikal markings explain procedures for entry and decontamination. No. Ah, a door we can cut our way through. While the main locks are open, the entryway is sealed with a hastily installed security iris. We should be able to cut through. Torch available. Prime. Ignite. If there's one thing I could say about this game that I would like to see it do a bit differently is the scale of time on which we experience things. The glass door here was violently shattered by the breach, breaking open what looks like a clean room. It seems like Dr. Vass actually experiences things in a much more accelerated way than we do, which is interesting. It's interesting. This looks like a suit fabricator, but retooled to build large-scale machinery. Its buckled plates reveal a crimson interior fused by rust. Like blocky coffins, these containers are still used by Baikal to ship their suits to active facilities. These are empty. Why are they empty? User, L. Bossmans. The Artificers. With them we could rewrite life as we wish. We could rewrite it to endure, Lucas, to surpass us. Okay, so the Artificers, which is something that we found out in the wild as looking like little skeletons, little coral skeletons, we thought. As it turns out, are artificial. Rows of chairs and corroded consoles scattered across the tabletops, precisely machine components thick with silt. Oh, for Christ's... Okay, fine, I'll tweet again. I did den to spell loading ready run right. Well, we'll probably have to scrub this run. Take the suit, Akari. Stop arguing. Without you, this project is dead anyway. Oki needs to survive. What's Oki? Nomura. My little Minnie, I'm sorry to leave you all alone. Sing a little for your mother. It's so quiet here. Holy shit. Her father? The lightweight lab suit sits propped in a chair. Sediment drifts from gashes in the soft material. Hollow sockets in the helmet faceplate. So what we knew of Minnie is that we worked on a project together several years ago. The suit is tagged Akari Nomura. I don't understand how. It's impossible. It's Minnie's mother. This is why Minnie came here. She was looking for her mother. She's been here a long time, decades. Has Minnie been looking for her all this time? God, Minnie, why didn't you say something? Anything. I can't... Give me a minute. While you need a minute, Dr. Vass, I'll, do some sonar surveying. The lab is ripped apart, plates buckled outward, massive coolant pipes breached, twisted metal now browned with age. Yes, that was where I wanted to go. Akari no Mura's lab. A long yellow cylinder marked with turquoise glyphs identical to the casing mounted on the back of the suit. Our suit. Wait, that's me! This is the same cylindrical casing as the one mounted on this suit. The same casing that houses you. Akari was building this, building it from artificers, building it with the machines in this arcology. She was building you. This is you. One of you, at least. Draft, an earlier version, a prototype. Akari is your mother, your creator. Did you know this? Did you know about any of this? Nuh-uh. Then you forgot? You malfunctioned? God, I can't think. We need to go. I don't want to be here anymore. Please. But there's another waypoint, Dr. Vass. The glass tubes that once fed coolant into the lab's core are shattered, glass shards now smoothed by soft currents. I want to go here, Dr. Vass. The inside of the pipe bulges with corroded growth. It stretches up towards the surface, and a faint circle of light glows above. Oh, it's a way out. The surface of the basalt towers dance faintly with the blue light that falls from the surface above. They look calm, ancient. Let's go have a look. The slopes descend into the dark ahead, their silver silt, a single plane leading toward an inevitable end. What is the inevitable end? No. We are out. I don't know what to say. We're still in range of the ROV on the slopes. Call for pickup. Let's go home. Anything else out here? No? Alright, Dr. Vass. Requesting retrieval. We're back. I'm down in medical. I disabled the quarantine. It's beautiful in here. Oki? Is that your name? I suppose it is. I like it, despite the history. Listen, I don't know what you're thinking right now, but I wanted to say I trust you. I don't know what you knew about the arcology, about Baikal's history on this planet. Even your own history. But I do know that you helped me, you protected me, you guided me. I owe you for that, no matter where you came from. You didn't choose to be created like any of us, but this place is your home. You were made from pieces of this ocean, from pieces of its past at least. I'm learning not to be afraid of the past. I'm not afraid of her either, of Monet. If that is her name now. I think she must have found something among those skeletons in the bloom, something truly alien. It changed her. It continues to change her, but perhaps that is what is needed. Change. I found something I think you need to hear. I've been going through the arcology data, and it wasn't the only facility on this planet. There were 2 other sites, 2 other Baikal facilities. We need to find them. Baikal was up to something on this planet, something bigger than just AI research. I don't want to look away. I want to see what Baikal did with my own eyes. I've marked the site's coordinates on the dive map. We should be able to cut our way in. This is not just about Monet anymore. This is about Baikal. It's about what happened here. It's about your history and the weight it carries. I'm ready when you are. Unidentified subject, DAT4 BR19CH. Well, that's bad. Ellery? Yeah, I really like Dr. Vass. She's, she reminds me of a lot of scientists that I've known, honestly. Curious, cool-headed. I know you have been reading these. The access logs shows someone has been in the file system of my terminal, and well, by process of elimination, it has to be you. I don't mind. In fact, I suppose I've been writing these entries for an imagined audience anyway. Confessions, or maybe just a need to talk. We make a strange trio now, a depressed xenobiologist who can't keep their mouth shut, you, an AI built from the remains of a sentient species, and your sister, Monet, who is now more plant than human. But as far as I can tell, we are the only ones on this planet who have any chance of finding out what happened here. I think I am starting to understand why Monet called me here. It was because she trusted me not to give up, not to let this go. Let's not disappoint her. Bical site, priority. Another Bical facility sits in the center of the bloom, beneath the Brocky shelf. In the arcology data, it is labeled Site 2. We have so many other things to, uncover. One of the Bical facilities was in this area. It is labeled Site 1. Was this the origin of everything that happened here? Though I'm wondering now, actually, if Dr. Vass uses they pronouns. Because in... where is it? Crew terminal? Who can't keep their mouths shut? I don't know. We have nothing more to discover about this molted skin, which is, a troubling thing to say. Analysis of the strange molted skin I found in the arcology cave shows it is clearly formed from the flesh of the slow masons. Within the tissue, the bodies of multiple polyps have knitted together over the span of decades, forming one larger communal layer. It may have provided protection, or perhaps it was the birthing sac for some larger unknown form. Shed skin can represent evidence of renewal or transformation. Perhaps the polyps are not mature, but are a larval stage of the species, with each member uniting to form a larger being. As of yet, I have found no trace of this matured form of mason, but the cave networks of the ocean floor could hide many creatures that could easily evade discovery. Who here has read Annihilation? It's a good book. But the slow masons remind me of just the way that we find them kind of attached to the walls, scrolling across the walls, leaving these patterns behind them. Reminds me a lot of, was it The Lighthouse Keeper? Or is that spoilers for the third book? God, I love the biologist. She was such a great character. It reveals the Lighthouse Keeper in book 1? Okay. Alright, well... let's dive. Now, we could also go out and just try to get, more pieces of things. which might not be a bad idea. Presumably these things exist so that we can find, like, good old chunks of things which make our lives easier. I feel like we're missing context still. Cold fire bathers, water bulbs, cold fire fans, deep orreries, snare veils. Signal stock tissue. Well, while we're here, let's try to get some signal stock tissue. Get the thingy. Chewy, chewy, chomp. Stock barrier. How do we get rid of these? Yeah, we need to find some shrill sacks. Bright pollen. Nope. Maybe we need to actually head back and go and get some bright pollen from, like, the starting area. And also drop off a bunch of our stuff. Is there anything in here that we haven't studied yet? I think we've done all of this. Oh no. Okay, maybe. Do we have any shrill sacks? What do they even look like? See, there's the sphere fragments. This is what Manet called the artificers, isn't it? Top right, or the one just under? Oh, there it is. Okay. We have a single shrill sack. So where are we going here? We need to get around this thing. Thingy! What is Thingy? Give Thingy. What are you? Soothe spores. Yeah, well, you know, I mean, while I'm here. No other way around, huh? Just straight out to the edge? Where do you want me to go for this game? This would be another point of contention I would have for this game, is that it's not always clear what it actually wants you to... Well, no, it's clear what it wants you to do. Doing it is tricky in a way that doesn't feel good. I guess. I don't know. You know, while we're in the neighborhood, might as well use the can. Okay. Shrill sack, get out there, do your thing. Huzzah! Broken boulder. Stress marks can still be seen along the edge of this huge rock, but it remains silent about its own history. The stalk looks silently out over the deep rift. I wonder if Manet looked over the same view with different eyes. Thingy. Grabby hands. Oh, good. More shrill sacks. Perfect. Chewy, chewy, chomp. This bubble-coated stalk has large gashes in its side and seems to be shriveling away. Its exposed tissue would be easy to sample. Well, let's go and get it then. New thing. Time to yell at some mushrooms. Sample candidate, exposed roots. Where the rock shelf has fallen away into the rift, the roots of the canopy above have been exposed. We could sample them from here. Well, let's go sample them from there. I wonder if we can actually get the other thing too. The one that's all the way, like, down at the south end of the reef. I think we have to go through here. Blub, blub, blub. Are we going across, or... Oh, right. Oh, can we not get back across now? I guess not. Interesting. Yeah, because we have no access to the stalks over there. We cannot yeet a soothe spore that far. Wait, can we use the booster to get across? I suppose we could try. What are you? Shrill sack. Handy. More soothe spores, also handy.
Oh my god, you guys get out of here!
More shrill sacks. Stock root. Well, we've already got those. We're in a totally different part now, I feel. Can I just say that I love the music in this game very much? More shrill sacks, that's okay. We're stocked. Couldn't eat another shrill sack if we tried. And we're all the way back at the start of the game. Well, at least we've got some things done. Oh, wait, no. There it is, sample analysis. A segment of tissue from a bubble-producing stalk. We unlocked the sketch. Reef stalks are fungal life forms which take the form of a series of stalks and plates anchored to rocky substrates. Their chitin exterior ranges in color from dark amber to acid yellow, leading them to resemble oversized lichens. Werewolves. This outer membrane is marked with slits which expand and contract, producing a distinctive hum. These slits or pores also allow both the release of spores by each colony and the absorption of other colonies' spores. I'm using this term for ease, but are these really seeds or something else? Keep drawing. Analysis of stalk spores has revealed a rich cocktail of compounds unique to each spore, some of which are previously unknown to human chemistry. This aspect of the spores and their noticeable exchange between the colonies of stalks suggests that these chemical cocktails are part of an information exchange network. I feel that we've read this. Right, stalk names. Oh, signal stalk, there we go. Tall, thin, bubble-coated stalks, right. Chemical analysis of a signal stalk tissue sample has confirmed the presence of a yeast-like fungus and large amounts of ethanol saturated in the tissue itself. This prompts a rethink of why many creatures target the hard-to-get signal stalks as a foodstuff. While I initially theorized it was due to their role as protectors of stalk colonies, the significant presence of ethanol in signal stalk tissue means most creatures would feel the effects of the alcohol consumed and may get buzzed from grazing on signal stalks. This, in moderation, would be a favorable, even enjoyable effect for the creatures, leading them to repeatedly target signal stalks. If I remain stranded on Gliese 667cc for long enough, I may need to make use of this particular biological property myself. Oh, now we have a sketch of the slow masons. Neat. I love how when you've completed something, you get to see it the way Dr. Vass sees it. Until then, it's just a signal on your sonar. But once you've done everything, then you can see it the way Dr. Vass sees it. All the way down here, huh? Yeah, because we made it all the way back to the central reef. The bloom is going to be a challenge. Ooh, I'm gonna take a quick break, and once we're back, then we'll do more. Sound good? All right. See you shortly, chat. I'm gonna run an ad as well. Hello, I'm back. Alright. So what do we think? Do we want to go and retrieve some of these things, or do we just want to progress the game? We also have a lot of, like, places we haven't investigated yet. Let's go and look for stock scraper shells. And then maybe we can figure out a way to get across here. It looks like there might be a way through here, actually. So let's see if we can figure out a way through there. Where am I in the story? We have investigated the arcology. And we have a line on the other sites. Use space jump. We also have a sister. As it turns out, the section of the shelf sheltered by the tall headland to the north is strangely peaceful. Well, let's go looking for things that we can kidnap.
Oh my god, guys, get out of here!
Why is Vector blocked? Beyond the thickly entwined stalks, a violent current can be heard. Oh, we need to use our space jump again. Stalk scraper. Ooh, a protected area. Little streams of sand pipe through the gaps in the rocks here, driven deep into the cracks by the current to the north. I like that description, sand displaying liquid behavior. Another rift separates the second and third fingers of the reef, looked over by a ragged stalk canopy that sheds particles into the flow. Strong current. Activate turbo boosters. And away we go. Wait. Have we already been here? Yep. God damn it, Nappa. Okay, back across we go then. There was a thing here. There was a thing I wanted to look at. Battered by the water, these stalks cling to the rocks. A single creature braves the current to graze on the twisted forms. Interesting. I have no idea if it was a desert or a Dragon Ball reference. It was something a friend of mine used to yell a lot. This canopy is partially bent by the current. If we are careful, we can sample a growth from its top. Dope. Let's be careful and sample a growth from its top. Three question marks. That indicates a high level of uncertainty. Object. These huge stones have been worn smooth by thousands of years of sitting beside the reef. Actually, let's, You guys, it's safe to come back now. White sand is gathered against the boulders on either side of this passage in soft, pale drifts. This stalk is torn open, perhaps by a predator. Inside, a rich tapestry of fungal life forms exposed to the open water are slowly dying. Well, we can help with that. Surely. I thought there was sampling available here. Alright. Back to Hombase. We were away for 3 hours. Look at all kinds of cool crap that we found out there. Annalise. Table stock. Intriguing. Sing stock. Attempts to analyze the interior of a sing stock reveals many different organisms within the stock that are not genetically related to it in any way. It seems that the sing stalks are actually tubular gardens which host juvenile fungal creatures. The swaying of the stalks keeps these sealed environments fed with filtered water and microbial life while allowing their spores to be distributed across the reef. However, I haven't found a single example of any of these life forms growing outside of a sing stalk, suggesting that they can only survive within these carefully controlled tube gardens, and that the spores they release are being used by the stalks for other purposes. Are these vertical farms designed to cultivate other species that might feed the rest of the stalks with showers of nutrient-rich spores? Neat. And table stalk. Laboratory studies of the growths found in the upper canopy reveals that they contain the genetic material of tens of different subspecies. These growths hold within their round casing seeds for many species local to the table stalks, as well as spores for the reproduction of the stalk colonies themselves. Fed by a carefully maintained flow of the correct minerals and chemicals, these seeds and spores are preserved by the stalks, kept viable for long periods. Could these seed banks be a complex example of collective serotiny? On Earth, some plants hold viable seeds until an environmental trigger, such as a wildfire or disease, causes their release. Perhaps these stalks are preserving the local ecology in case of environmental disaster. Are they the guardians of the reef? Neat. Maybe if we collect enough stuff, we can help Minay. What were the things that allowed us to traverse the blight? They were the nest workers, right? Wasn't there something else as well that attracted the bivalves? And the bivalves would create little zones? I want to say it was these things. Because we'll need to go to the, Oh no, wait, hold on, there's more things to discover. All the way down here, huh? Was it fan dust? Fan dust sounds right. But look at all the things that we can find. We can help Minay maybe if we discover all of this. Maybe we can get the good ending. Alright, let's see if we can get, well, let's get the stalk scraper and see if we can get down to wherever this is coming from as well. Sorry, I'm just kind of like in derping around mode today. You know, we just found out that we have a sister. It's big news. It's big news. Yeah. While we're here, you know, might as well get one for the road, you know? Might as well get a shrill sack for the road. Oh wait, there's another one down there that we totally neglected. This is the way to the thing. This is how we get to the thingy. And a thing we haven't seen yet. Bloated stock. When alarmed, these stalks burst open, releasing their toxic contents into the local water. Oh, they're tweets. Well, let's try not to alarm them then. Hello, what's your name? Bloated stock. Something is happening inside you. Ah, God, it got on us. It got on us. Ew, ew, ew. Ugh. It peed in the pool. Okay, they don't like shrill sacks, as it turns out. Those things do not like having shrill sacks go off next to them. To the west stands the final pathway of stalks before the colony fades out into the sand and rock. Reef blowout. Large bloated stalks that burst easily. That's enough data to log and classify these inflated stalks. I'm going to call them reef bloats. Their movement suggests unseen processes. Ha! Twofer. What's out here? Way down. I don't think we've been here before, have we? Or have we? No sonar pings, so I don't think we've been here before. We must be heading beneath the stalks. That's biological. Thick, bloated roots poke through the rock wall and something pulses within them. This is a rare chance to sample the stalk root network. Let's sample the stalk root network. What was our name? We had a name. It turned out that we had a name. Our mother gave us a name. Oki, right. What manner of name is that? I'm assuming it's Japanese. But what does it mean? It looks like we've been here already. Let's call for pickup. Open sea? I like that. Let's see, where's the... Oh no, we need to actually transfer the samples to the thing. Bloat root. Reef bloat taxonomy entry updated. Reef bloats are large bloated stalks that can be found at the western border of the reef. Intimidating in their size and distorted shape, they resemble inflated sporing mushrooms covered in nodules of rampant fungal growth. Their membrane, which is stretched by the bloating process to an almost translucent sheen, is darker than a typical stalk, making the stalks themselves a shade of sickly orange. What role the bloat plays in the reef stalk now? Oh, so they're like windowed, like well-developed bread dough. What role the bloat plays in the reef stalk's network is not immediately obvious, but their pulsing pumping motion suggests that the bloats are either processing or producing the toxic microbes that are contained within their fragile membranes. Without analyzing the microbial colonies themselves, this will be difficult to ascertain. Finding a root sample from a reef bloat has brought with it the confirmation that reef bloats are not manufacturing spark colonies, but instead modifying, metabolizing, and processing them. Analysis of the bloat root system has shown the presence of microbes at various stages of modification, including those that had been entirely detoxified and their rampant metabolism neutered. If all reef bloats are connected via this root system, then they may be part of a large-scale cleanup operation with the aim of eradicating these aggressive microbes. Reef bloats are not diseased stalks then, but powerful biological engines which are fueled by the local stalk colonies. The scale of this operation suggests that, for the stalks, the spark microbes are a significant threat. Neat. So the reef bloats are actually like... Well, it's very Nausicaa, isn't it? Oh, Dr. Vass is just sitting out here in the observation blister. That sounds nice. How's Monet doing? Growing healthy. Now it wants more. Stalk scraper shell. So where did we get to? I think we were all the way over here, weren't we? This is where we found the reef bloats. Near the bloom. So we need to go southwest. Right, because the initial waypoints take us west. Or wind up taking us west. So we need to hang a left here. It's weird that we can't just go south. Maybe we should try another direction from the main base. You would think there would be a waypoint down here. Well, might as well load up while we're here. Yeah, this looks promising. I mean, we've clearly been here before. But I suppose we turned around? Well, I mean, maybe we should pick up some shrill sacks for the road. Ba 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 ba. Ba ba da ba da ba 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 da ba. Bum 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 bum. I forget how the rest goes. I think they were... Oh, you know. This might be the way through to the other thing. The other thinger. The other thingy. What are you?
What are you? I don't even remember what you do.
But I like stuff. Oh no, we frightened it. Ah, yes, we definitely seem to be going towards the thing. Oh, right. Right, okay, this is the way we came before. I'm dumb. So this may be where we're able to find, the thing that... other thing. The thing. I'm not lost, but I am aware that we are just kind of like retreading old territory. I wish there was some kind of overlay you could bring up that would show you like where you want to go and where you are in respect to where you want to go. Some kind of world map? Like, I realize you can see further than what's just within your thing, but, like, the thing is, the thing doesn't help you find the thing when you're looking for the thing. You know? Is this where we just were? Okay, like at the base of this finger, right, of the thing, seems to be where that one sample could have been taken, right? Turbo boosters activated. Ew, greasy streaks on the helmet. It's like a bus window. Aha! We're full. Oh. Well, what if we throw away a shrill sack? Goodbye! Run free! Chewy, chewy, chomp. More lore, please. Analysis of reef cap tissue shows that the reef caps maintain a bacterial symbiont within a series of internal chambers, which, while unique to Gliese 667cc, resembles Epulopiscium. A large-shelled bacteria found in the stomachs of terrestrial surgeonfish, Epulopiscium. Epulopiscium helps surgeonfish break down algae effectively, and the reef cap's bacteria seems to do the same, with traces of an algae-like growth found within the cap's chambers. Where is this algae coming from? However the caps receive it, this algae is allowing the caps to grow bacterial colonies, which then reduce themselves to endospores when the algae is digested. The endospores are then released to the stalks in the local area to renew the strength of their colonies. It's an incredible recycling system for protecting this ecosystem. Sketch. Neat. I would love... do they train biologists to do this? Because they just told us to use ChemDraw, and ChemDraw sucks. Okay, we're collecting all the things. Eventually. Coldfire bather root, snare veil tissue, deep orrery tissue, cold fire fan tissue, water bulb tissue. Let's go for a brief change of pace. Abyssal sail shell. Let's go deep. We never did go this way. Okay. We seem to be through. No more veils ahead. After the last veil, the dark returns. Creating bright folds in their gossamer material. Veil field. The tangles of veils close around the outcrop here, encasing it in a prison of glinting light. At the edge of the light from the fan, the petals are pale and twisted away, perhaps turning to another source of food. Surrounded by its audience of light-bathing petals, the fan's sheath shimmers with amber light like a bonfire. Ooh, sampler available. Well, we'll take these in case we need them. Among the elegant shapes of the petals, small, stilt-legged creatures stride, cleaning particles from their pitted surfaces. More samplies available. It's a Greek word. Deep pollen. Well, we'll not take any, because we're getting full. We couldn't eat another bite. Twisted back on itself, this veil is being eaten away by creatures, its defensive abilities neutered. We could sample some tissue here. Yes, please, let's. Cool. We're learning about things. We're doing science to it, Chad. Having sampled a dying veil, analysis of the tissue demonstrates what a unique creature a snare veil is. Its tissue is threaded through with powerful neural connections, lined with a digestive surface like the interior of an animal intestine, and coated with a layer of light-emitting cells. It is as if a single creature had been turned inside out and rolled out until it was a flat plane, millimeters thick. A snare veil is a totally distributed form of life with no centralized brain, stomach, or other organs at all. It is self-same in its entirety. Any other piece resembles another. What we saw as a dying veil might have been purposefully separating itself, as each piece of veil could grow another tangle. No wonder they've dominated portions of the seafloor. Neat. Okay, so stalk scrapers and lung crests are still unknown to us. But we're getting there. So we got this one. But if we just go due south, we should be able to get to a node where we're able to find a lot of other deep things. Hopefully this will let us help Minay. I hope I'm pronouncing her name correctly. Ooh, shell segment. Gib. Another piece of territory yet to be claimed by the strange pattern of the veil field. Two veils that look as if they have recently split present a narrow gap in this Ow! What the shit? Get thee away from me, thingy. I don't like you. The veils meet here, but through their translucent skin, I can see the dark shape of an outcrop. Or... Ah! Oh, this is gonna suck so bad. Ow! Dr. Fast, why? You has betrayed our tiny trust. I think this burn that I got making bread last week is gonna scar up nicely. Shed polyp. Ooh, the deep orrery. Having analyzed some tissue from the orrery's agile prey, we can begin to understand their behavior. Unable to match other creatures for speed, orreries seem to rely on the dangers presented by deep-sea veils, beside which they are often found. Veils funnel cautious creatures into the orrery's nets, while the orreries themselves don't seem to be affected by the veils and are able to traverse them easily. Perhaps the two have a symbiotic relationship, hunting together in a mutually beneficial manner. Outside of this, orreries appear to be mostly lone hunters, and I have seen no social interactions between individuals. But what exactly is an orrery? I would like to understand more about their strange, uneven forms. Perhaps a polyp that had been shed might provide details on their anatomy. The only lab injury I got, and it's faded now, was the time I got frostburn from decanting, well, not decanting, from pumping liquid nitrogen into a big dewar flask, because I was holding the nozzle into the flask, And as I began to pump the liquid nitrogen in, I realized the glove had a tear in it between where the thumb joined the palm. And so as I was holding the brass nozzle, it got very, very, very cold, and I wound up with a burn from it. But I couldn't just let go, right? Because that would wreck my shoes. Analysis of an orrery polyp has demonstrated that they are indeed colonial creatures. They appear to be made up of both specialized animals of the same species and different creatures of different species. Based on this, the orreries may need to be reclassified, although that would require extensive study and equipment I don't have access to. What I can say about the orreries is that each one seems to have specialized its own growth based on its requirements. Many of the zooids that are part of the colony are able to shift between medusoid, dactylozoid, gonozoid, and gastrozoid forms with incredible speed, depending on whether the colony needs to focus on propulsion, feeding, defense, or digestion. This makes each orrery a complex social and biological structure, like a floating city rather than a lone hunter. Cool. So we got the one here. We got the one here. Let's head back out then. Actually, I'm going to... The odds that we're going to need any of these things are vanishingly small, and maybe we can just get the entire thing done. Maybe if we classify all the life forms on this planet, we'll be able to help Minay. So there should be one here. No touchy. There's an orrery. What are you? Digested remains. Yeah, we can use this to feed our suit. Or feed ourselves when we run low on power. We've been here before. Deep pollen. Not interested. What are you again? Fan sheath. 30% mucal. That's what they called me. Maybe the Veils don't like the outcropping because they can get smashed against it or something. Okay, so what we're looking for is not around here. Just to be sure. Ah, what are you? No, it's okay. I'll wait for you to leave. Maybe if I yeet this at you? What are you? Among the hypnotic patterns of the veil's other lights move pinpricks like LEDs shifting in the loose orbit of an unseen body. Attract the player with light. Can we get there from over here, maybe? And not have to go through the veil? No, we have to travel through the veil. That sucks. Abyssal sail shell. A small dark creature with a large translucent tail shell that allows it to rapidly move through the water, navigating the veil tangles safely. I'm calling these abyssal sail shells. Those wide tail plates are instantly recognizable. No, no, get away, get away, get away. Go away. Not food. Strange thing. Is this our first question mark of this expedition? Or this particular expedition? No more fan sheaths. Okay. Looks like we've already been around here. Ow! Yeah, there's another thingy down here. Oh, ow, why? Oh, it's a thing to sample. Okay. All right, let's get out of here. I'm just kind of enjoying this, I think. Going around, learning about the planet, doing science to it. Or enabling our human to do science to it, more appropriately. Oh, new taxonomy. Animalia. Anthropelta. Abyssal sailfish. Sail shell, excuse me. These shelled creatures with their large translucent tails are surprisingly fast for animals of their kind. Occasionally spotted weaving between veils in the deep, they use the speed as a way of evading the slow-moving tangles. They are also dark in color and small, making them a difficult target for other creatures that might hunt them. They use modified back legs with wide oar-like shapes to both propel themselves and make rapid turns, but most distinctive of all their features is their large translucent tail, which stands like a crest behind them. From my observations, these tails seem to be a form of propulsion for the sail shells, but until I can inspect a fragment of one in more detail, this cannot be confirmed. Tail segment. Water bulb. Oh, so many things updated. After finding a segment from a sail shell's tail, it seems obvious that these creatures use their tails for propulsion, but rather than flapping them like a flipper, the sail shells use their tails to catch currents and allow the flow of water propel them through the deep ocean. Their tails and the rest of their shells are incredibly light, which means that even a mild flow is enough to grant them significant speed. This may also be why they follow the same paths. Their propulsion is best suited to using currents where they can find them rather than choosing their own route. But why travel back and forth through dangerous veil territory over and over? Perhaps a look at a full sail shell, even a larval one, could help us understand this. Analyzing a water bulb bud has provided an important insight into their anatomy. The bulbs themselves are mostly hollow with only a thin membrane filled with pores separating them and the water around. The interior side of this membrane is iridescent, lined with hexagonal crystals of guanine, producing an effect like eye sheen seen in terrestrial cats, sharks, and other creatures, but on a much larger scale. These crystals reflect light onto the sphere at the center of the bulb, which is in fact a huge and impossibly complex compound eye. This eye can see a broad spectrum of light, including UV and IR, with at least 36 different photoreceptor types. What are these water bulbs observing, and why then are they so passive? Just the thought of their total vision makes me nervous in their presence. Cool. Typos are interesting because they usually occur where somebody has edited something. Like, where was it? Can see unbroad spectrum of light. There is probably another adjective in here, beginning with a vowel. Yeah, an incredibly broad spectrum of light. And that adjective was deleted in an edit, but the word was not changed. Cool. Having analyzed some tissue from the orrery's agile prey, we can begin to understand their behavior. Unable to match other creatures for speed. Okay, right, yes. There's so much to learn. All right, we're gonna go out once more to the depths. Oh. Let's see if we can finish sampling all of this stuff. We're gonna get that good shit. They want the cold fire fans. Is there anything out here? No, there wasn't. Am I completely lost? I thought the thing that it wanted was out around here somewhere. But these are just... I mean, I suppose we can grab it. But I don't think that's what she wanted. Okay, and this is where we want to go and find the sail shell eggs. Or a nest, maybe? I feel? Ovular growths? This may be the eggs that we're looking for. These may be the eggs that we're looking for, plural. But maybe not. Who can say? Who can ever say? Who knows? It's impossible to know one way or the other. See, you stopped there. Why did you stop there? Aha! Animal egg. Well, might as well just throw ourselves through this net of stinging cells. Oh! I've become digested. Aha! A wilted fan faintly pulses, while spines of its body have broken away, still faintly glowing in the silt. We could sample them here. Good, yes. Oh, I'm full. I couldn't eat another bite. Alright, let's get out of here. I think we found enough to fully classify 2 more organisms. Glowing spine. Cold fire fan taxonomy entry updated. Abyssal sail shell taxonomy updated. What are you? Petal root. Cold fire bathers taxonomy updated. I'm always... Some things become Okay, but we already knew about the reef bloat. No relevant taxonomy entries found. Weird. Is there any more we can learn from analyzing these things out of order? Okay, the brine remains mysterious to us. After analyzing a clutch of beaded sail shell eggs and inspecting their anatomy, I've started to understand their behavior. Larval sail shells are already forming modified limbs within thin, thread-like whisks on them. These limbs move so fast when the sail shell is swimming that they can't be seen, but in the dormant eggs they are clearly visible. These whisks would allow the sail shells to feed by catching small larval creatures, as well as any other particles of nutrients found in the water. This must be what the sail shells are doing when they follow their carefully selected paths back and forth through the waters. It is also likely that the veils attract, trap, and produce tiny particles that the sail shells feed on, hence their close proximity. The results for sail shells is a very risky feeding pattern. Neat. Analysis of the cold fire fan spine we acquired shows that fans not only possess light-producing cells, but also light receptors too. These incredibly sensitive cells are found on the front of the fan and show the fans are able to perceive the lights of other creatures, perhaps including other fans. As each fan tends to its own oasis, perhaps the fans use their light as territorial markers, signaling to other fans that their basalt column is taken. They may even use them to communicate with other species, responding to the flashing patterns of veils, for example, and letting them know not to approach their territory. Like lighthouses, cold fire fans seem to act as both safe havens and warning signals, depending on the creature they are signaling. Yes, I thought we had this. Laboratory analysis of the microbial colonies found within reef bloats, spark colonies as I have come to call them, shows that these microbes are not of a group typically associated with stalk colonies. In fact, spark colonies appear to be made up of highly modified algae with a wide metabolic range, able to consume carbon dioxide, oxygen, metal, and toxic compounds with equal ferocity when supplied with a source of light. When metabolizing these substances, spark colonies produce a regular flow of charged electrons, making them ideal for use within a microbial fuel cell, or MFC. Their suitability for this use is suspicious. Are these really naturally occurring microbes? And why are they found only within reef bloats? Something isn't right here. Oh no, what did we do to this place? The sample we acquired of a bather root network shows the presence of bulbs within their root network. These bulbs appear to be a way of the bathers storing the energy gained from their photosynthesis of the fan's bioluminescent light. Concealed in rocky cracks in the softer silty substrate of the seafloor, they must allow bathers to outlive fans which die off or stop producing light. This suggests that despite appearances, it is not the fans which maintain the bathers, but the other way around. The bathers then, despite appearing like worshippers of the fans clustered around their bases, create the conditions for the fans to grow within, ensuring themselves a regular supply of light in exchange for oxygen. In a sense, the fans are their captives. Cool. All right, I'm going to take another break, and then we'll be back. And if you're good, I'll advance the story, chat. All right, back shortly. Hello, I'm back. All right. What else do we have left to discover? We've managed to clean up all of the deep things over here. But we still have some stuff over in the brine pools. And The stalk scrapers. We have yet to clear up the stalk scrapers. Brine pool time. We never did explore over here. Maybe this is the way we're supposed to go. Maybe this will lead us north. Aha! Deep pollen. I forget, what does the brine eat? Is it our power or oxygen? Oxygen? Okay, handy. What are you? Well, this one looks dangerous. Let's go over here. Ow, 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 ow. A thin causeway leads away from the lake. Unlike the shallow seep marks in the south, these deep craters plunge through silt into the bedrock. The searing pain. These aren't seep pools. Something impacted the seafloor here. Meteors? But how could they get so deep? Or could some creature have left these? Ah, we did some sequence breaking, apparently. Along the edge of the larger pools, the shells and skeletons of creatures loom out of the silt, victims of the toxic brine. A bank which skirts along the edge of a large shadowed pool. Okay, we've been here before. Oh! These are the microbial mats. Yeah, brine mat. Okay, well, I mean, they're handy at least. We can eat them. But there's nothing here that we haven't discovered yet. So let's go back and then dive again. Nope. No, still the same. This feels bugged. I thought there was something that we got. Apparently not. Yeah, we need to just go basically due north through the brine pools. Maybe it's where that ship crashed. I guess we missed some stuff through there. What are you? Brine Strider. Oh. I'll call these mournful creatures Brine Striders. They're walking lanterns calling out in the dark. Very good, Dr. Vass. A split in what must have been the vessel's hull allows passage into the overgrown interior. Sounds good. So what am I missing through here? Most of the wreck must have already been lost to the brine, only this section remains precariously settled on a silt bank. What is the brine, though? Is the brine naturally occurring, or is it something that this ship spilled? Could this set of seeps have been created by the wreck's impact? Perhaps it breached the bedrock, protecting the methane below. Oh. A crested shell sits on the edge of the brine pool, its former inhabitant nowhere to be seen. We could sample it here. Oh, this is gonna suck. Oh no, wait. There we go. We're fine. We're frosty. We're hip. We're happening. Let's get back out of here. All right, then we'll head the other way. Maybe it's over here. Because we've been through here before, but I feel like we just discovered what we were looking for and then left without really exploring it. Yeah, methane clathrates. Because the brine isn't really like, it's not, I'm assuming it's not sodium chloride brine. This is some kind of toxic salt. Or, like maybe a metal salt of some kind. A reducing salt? That would make sense, it eats our oxygen. Actually, I don't know if that makes sense or not. There we go. I think that's a way through. Ow. There we go. There's something we hadn't looked at yet. Fronds crowd this piece of debris, some eaten away by the local grazers. Broken limbs lie all around. We could sample one of the smaller pieces. What are you doing? Who are you? What are you doing? Animal and tissue. Looks like we got there, chat. Anything else? No? Let's get the hell out of here. It would be nice to be able to tab through this by environment. Bristly limb. Brine eaters. Lung crest. All right. Lung crests resemble large crabs in their basic anatomy, though they are instantly recognized. Okay, right. Sampling a lung crest shell has made the function of their mucus-lined bubbles clear. After seeing lung crests scavenging around and even entering into the brine pools, it seems obvious that their bubbles are in fact natural scuba gear, allowing them to move around the anoxic environment of the brine lake. The shell confirms this with a clear vent in the shell, which allows the lung crest to access the gas within the bubble without breaking it. This unique evolution means that despite their slow speed and sheer ignorance of what goes on around them, lung crests are highly resilient, with their thick shells and bubble crests together forming a kind of analog of my own dive suit. I'll be sure to say hello to these fellow divers the next time we visit the brine lake. Brine eaters. Inspecting the tissue of a brine eater shows that despite a full nervous system, they have no central brain. In fact, regeneration present in the tissue sample suggests that a brine eater could be regrown from just a single piece of an individual and is highly resistant to predatory damage. With no central self to orient themselves around, brine eaters are a highly resilient form of life, which is perhaps how they have come to exist Was this dictated? Which is perhaps how they have come to exist in one of the harshest environments of Gliese 667cc, the shore of a brine lake. They are so resilient, in fact, that I would expect that brine eaters and their variants might be found across this ocean, each one grown from the smallest fragment of one original individual deep in the past of this planet. Cool. That should be everything down here. Except for this. 7 crabs. Oh, this sucks. Okay, fine, Dr. Vass. Fine. Fine. We'll go and get it. No, I don't mind being dissolved at all. Why would you ask? Okay, so we have to cross this huge brine lake. Mucus bubble. Ow. Dr. Vass, why do you do this to us? More mucus bubbles. In fact, I think I'm going to have one. Glowing shape. From this small pool at the lake's edge, a pale blue pulse is flashing. I swear I see lights blinking in response out above the brine lake. Well, let's go and investigate. Maybe this is what we're looking for. Ow, it's eating us. Brine shell. Oh, a young carapace. Is this what we were looking for? This must be what we were looking for. Well, let's get out of here, and if it's not, then that's okay. I thought we already had this, though. Dr. Vass, you know we have a couple of these in storage, right? We found these already, and we gave them to you. And you were like, boring. I swear to God, Dr. Vass. I swear to God. I swear to God. You do this to us on purpose, don't you? Brine striders are towering crab-like creatures, right? Yes. Keep their carapace from the toxic waters. The bones and shells of creatures that die to the brine pools adorn the strider's carapace. Pulled from the brine by a pair of extended claws, among these rocky piles, smears of bioluminescence glow blue. This pulsing life, whatever it is, finds shelter in the remains of the striders collect. But why did the striders make these creatures a home in the first place? I am also curious how, if the striders are vulnerable to the brine, do adolescents reach the heights required to survive? In order to understand more about these obscure creatures, perhaps we need to analyze the glowing, brine-clogged remains they gather. I just realized, of all the games this reminds me of, it's Alpha Centauri. This reminds me so much of the way that the Gaian technology, the Gaian's leader, the way her voice lines were structured in Alpha Centauri. Analysis of a brine-clogged shell reveals a complex chain of chemical reactions. Among this chemical richness, a form of extremophile bacteria feeds on the brine's chemical traces. When the striders collect these remains, they also collect the bacterial colonies which live on them. Removed from the brine, but still feeding on the chemical-saturated shells and bones, the bacterial colonies produce a bright glow, a side effect of their metabolism. Do the striders know that they are turning themselves into pale beacons? What is the nature of the relationship between these bacteria and the striders? I need to understand more about the life cycle of a strider and how they grow to such heights. Analysis of a juvenile strider's carapace paints an enigmatic but beautiful picture. The adornments of shell and bone only begin as the brine strider matures, each arrangement being the life's work of that individual. Though the striders often live apart from each other, it seems that they still communicate. Their adornments, which grow taller and brighter, act as beacons. They can signal for food, signal to mates. They keep company from afar. Gathering the remains and their glowing bacterial colonies is a ritualistic process, a way for each of these large and ponderous creatures to create an identity within the ecosystem through a living, growing work of biological sculpture. Cool. What more? You want more? We had it all. Dr. Vass. We were done. Very well, Dr. Vass. I would be happy to gather more samples for you. Oh. Right. Yeah. We need to go and collect some crab. We need shrill sacks. Where can we mine for shrill sacks? How do I mine for fish? Chewy, chewy, chomp. Chewy, chomp. Chewy, chewy, chomp, chomp. I swear, Dr. Vass, if this is something that we already have in storage, I will be completely powerless to do anything about it. Carrot, carrot, milk, carrot, milk, carrot, milk. I was truly not expecting James to come up with this summer's seasonal banger. What were we even looking for here? Oh, maybe it's this thing. Stock bark. There's at least one sample you'll need to advance the story to get. I mean, That does make sense to me. No, we've been through here. We've been through here, Dr. Vass. Not this time. Ah, 7 craps. Already? We just threw away Stock Park. Great. You are a shrill sack, not the thing we were here to collect. Through a biochem bachelor? The thing that helped me get through biochem, because I had to take 2 semesters of biochem, was learning to accept that when they told me, don't worry, you don't have to know the mechanism, you just have to memorize the reaction, to just accept that. That was the biochemist's way of saying this mechanism is not well understood. Because as a chemist, that was the kind of shit that drove me up the wall. It just completely shattered my will to go on when a biologist would tell me, oh, don't worry about knowing what's actually happening here. Just memorize it. Because as chemists, we are taught to rationalize and remember things via their mechanisms. And so every time I was told, just memorize it, I died on the inside. But like, if they had just said, oh, the mechanism isn't well understood, that's something we get, right? That's a thing by chemists here frequently. Oh, we don't know why this works. Okay, that's fine. People have won Nobel Prizes for poorly understood reaction mechanisms. No, admitting you don't know everything is how you learn things. Eventually we'll stumble ass backwards into whatever the game wants us to look for out here. Aha! Eth. I don't know why I added the ETH there, but it felt correct. A small clearing between the avenues of stalks. To the west, their ordered rows dissolve into the chaos of the southern forest. What are you? Spore catcher. I'm going to call these spore catchers as they are so eager to gather the stalk's spores. Not just a clever name then, Dr. Vass. What are you? Question mark. Animal. Shell. Part animal and part shell. See, I always joke that I went into theoretical chemistry because I was shit in the lab, but I was actually pretty good in the lab. I kind of miss being in the lab. Oh no, this is the thing that's going to get mad if we use the shrill sack near it. Oh, wait, we haven't gone this way yet. One ping only, please. No, wait, we have. We have come through here. We've absolutely come through here. I'm beginning to become disenchanted with this process, Dr. Vass. Yeah, I mean, I probably would have been less irritated with the physics students deciding to graffiti on our whiteboards and blackboards that we didn't know anything if most of their advice wasn't, well, why don't you just abstract it more? You have all these like terms on the end of your equations which are not useful to you, so just delete those, and you're like, those are empirical terms, those exist in real life. We have to deal with things that you treat as non-interacting spheroids. We actually have to deal with those, you know, because we exist in 3 dimensions. We don't get to just like erase those. You dumb bastards. Okay, stock scraper. Finally! Finally! Spore catcher. Behavior. Analysis of the unique... Oh no, wait. Let's... Observations. Spore catchers are small, soft-bodied creatures which move quickly through the water using siphon jets. Their mantles, striped with bright colors that shift as they swim, allow the creatures to modify their movement through the water with precise flapping motions. Watching them navigate the water is hypnotic, their rapid turns and loops showing incredible agility, flitting through the water like a bat or bird. Spore catchers use this wide range of movement to pursue the various fungal spores that are produced by the reef's stalks. The spore catchers seem to be highly selective about which spores they wish to consume, suggesting that analysis of the stalk spores might tell us more about the spore catcher's behavior and diet. Analysis of the unique chemical compounds found in stalk spores and a wider understanding of how stalks use spores to communicate suggests a more mutual relationship between the spore catchers and stalks than initially assumed. If stalks are able to tailor the chemical makeup of their spores, it is possible that the select spores the catchers eat are intended for their consumption and contain compounds and minerals attractive to this species. As spore catchers use their color-shifting properties to hide among stalk colonies, it seems increasingly possible that stalk colonies and spore catchers are mutualistically linked, and catchers serve the needs of specific stalk patches. What those needs are, however, is not yet clear. Analysis of a spore catcher egg, if found, may help us gain a full picture of their physiology. I wonder what an alien biologist would make of a human feeding ducks by a pond. It's unclear why the subject is surrendering part of its own diet to these other animals. There's no clearly mutualistic behavior here. Egg sample. We're gonna have to go out again. Chemical analysis of a stalk scraper shell confirms that not only are they made from stalk membrane materials, their distinctive cloudy coloration comes from stalks too. Stalk coloration, like most fungal coloration, is reflective of the local water acidity and the medium the fungus grows in. This variance of amber, orange, yellow, and even green pigments is passed directly to scraper shells when consumed, resulting in beautiful whorls and blobs of color. A scraper's shell is a living record of every stalk colony it is fed on, and therefore a marker of territory and social standing. Scraper shells could be used to help study the entire reef and its shifting ecosystem, each one a chemical map of the local history, readable for those who can learn the significance of its forms. Cool. Ah! Spore catcher egg. Fine, you get one more, Dr. Vass. You get one more. You get one more. I remember the first time I had a physics prof tell me that you could multiply both sides of an equation by dx. And I was just like, oh, I get it now. You're full of shit. This entire discipline is full of shit. You're not magic at all. You just make things up. I feel that what we want is up here. There we go. Anyway, it was then that I realized that I refused to be condescended to by anyone who multiplies both sides of an equation by dx and goes, done. Math understood. Animal egg? We did it, chat. By which I mean I did it. And you were also here. This is the part where the physicists explained to me that actually multiplying both sides of the equation by the dx represents a much more sophisticated understanding of things. Just tell me you're shortcutting. Don't tell me you're multiplying both sides by dx. Oh my god. Being like, this is when we paid someone from the math department to solve this for us. One of my profs was like, there's a whole building over there of people who do this. Just get one of them to do it. They'll happily put their name on one of your papers. Make sure you follow along so you know what they're doing, but go. Doing the integral yourself is a sure way to screw it up. Analysis of the spore catcher egg shows a strong correlation between the minerals it contains and the minerals present within the catcher's favorite spots. It seems that through the spores, stalks are able to affect this catcher's egg production, therefore controlling the volume, traits, and behavior of catchers within any area of the reef. In a sense, the stalks have domesticated the catchers, leaving the two intrinsically linked as species. But the question of what the catchers provide the stalks remains. Do catchers serve a specific purpose, consuming unwanted or incorrectly configured spores, for example? Or are they kept for other reasons? Could the catchers be kept by the stalks for their companionship and social use, just as humans keep pets? Oh my god! They feed them just the same way that we feed ducks! I was right! I was right! All right, I'm gonna thank subs now. But wow, we cleared out all of that stuff. I had a math prof who actually structured, or my partial differentiate, my PDE's prof structured his assignments to goozle Wolfram Alpha. He'd be like, I can tell which ones of you just plug this into Maple or Wolfram Alpha because your solution is 12 pages long. If you had done this by hand, it's 3 lines. Alrighty. Let's thank subs, shall we? Original Garwolf, sub for the 40th month, saying this sub brought to you by the Organization for Social Distancing. If you can read this message without glasses, you are too close. Ivrl has subbed for the 8th month. Lady Lockwood 92 resubbed for the 3rd month, saying I really love this game. It's such a beautiful experience. Thank you for bringing us along, Cam. Well, thank you for having me. Deathrite Shaman resubbed for the 15th month, saying whoop, it's been 3 times 5 months. Charlemandius resubbed for the 26th month, saying, the soothing tones of Cam to accompany me during work. Blind Prophet 32 resubbed for the 38th month. Thagnock resubbed for the 12th month. Happy 1 year. Archonic Energy resubbed for the 44th month, saying, oh, look, a button. Must press it. Vegan Troll resubbed for the 21st month, saying, oh, a cam stream. Always a good time to sub. Also 21 months. That's how long we'll stay isolating. What a coincidence. It's Eros resubbed for the 15th month, saying, how are you doing, Cam? Yeah, all right. I'm doing all right. Atomic Alchemical resub for the 9th month, saying, always a pleasure to tune into Talking Sim. Listening to Cam is so very soothing. CF Preston resub for the 38th month, saying, stuff! Admiral Albatross resub for the 4th month, saying, bird noises. Tuples, or perhaps tuples, if they're Greek, resub for the 49th month. Lapolio resub for the 25th month, saying, no more fans. Affinity for Aleph resub for the 4th month. Lady Lockwood 92 cheered with 250 bits, saying, really enjoying your playthrough, Cam. It's super clear how much you're enjoying yourself. I kind of love this game. It's good. Consider purchasing it. Insane 42 resub for the 14th month, saying, hi Cam. Hello. Zoj 333 resub for the 3th month, saying, hey, missed the start, but I always wanted to say I've been really enjoying In Other Waters, as Cam and Corey are a delight. See you in the VOD. To Spoiler 98 sub for the 11th month, saying, Cam, hope you and the crew can stream together in the same place soon. Thanks for all you guys do. We're hoping. The numbers in BC are good. They're very good. Maybe soon. Maybe soon we'll all be able to get together again. The File Clerk resub for the 25th month. Thank you. Iris of Ether gave us 100 bits, saying this is a perfect soundtrack to my document editing. Thank you. Flickers 1 resub for the 60th month. Happy 5 years, saying, I missed a lot of the early parts of the stream. What was the consensus on the AI's personality? Does it think? Sorry if this has come up a lot. I think it's a lot like my own. I mean, honestly, it is the player character. So its personality is largely inveigled behind a forehead. Duckydale 594 resub for the 10th month. Thank you. Etheroslovek gave us 100 bits, saying, hi Cam, popping in to resub with my Bezos bucks and sharing with my last actual bits. Hope you and the rest of the crew stay well. Thank you very much. I hope the same. And Coreflux gave us 100 bits, saying, this is such a relaxing stream, so chill. How does this game resolve the in-universe China room problem with the AI? I'm not familiar with that problem. Maybe I can look it up. I suspect it resolves that problem by ignoring it. China room problem. Thought experiment of John Searle and associated derivation. It was the best known and widely credited counters to claims of artificial intelligence. That is, to claims that computers do, or at least can, someday might, think. The argument is based on two key claims. Brains cause minds, and syntax doesn't suffice for semantics. Its target is what Searle dubs strong AI. According to Strong AI, Searle says, the computer is not merely a tool in the study of the mind, rather that the appropriately programmed computer really is a mind in the sense the computer's given the right programs, can literally be said to understand, have their cognitive states. Searle contrasts strong AI with weak AI. According to weak AI, computers just simulate thought. Their seeming understanding isn't real understanding, just as if. Their seeming calculations is only as if. Calculations, etc. Nevertheless, computer simulation is useful for studying the mind. Again, strong AI asks you to imagine yourself in a monolingual English speaker locked in a room, given a large batch of Chinese writing plus a second batch of Chinese script and a set of rules of English for correlating the second batch with the first batch. The rules correlate one set of formal symbols with another set of formal symbols. Formal or syntactic, meaning you can identify the symbols entirely by their shapes. The third batch of Chinese symbols and more introductions in English enable you to correlate elements of this third batch with elements of the first two batches and instruct you thereby to give back certain sorts of Chinese symbols with certain sorts of shapes in response. Those giving you the symbols call the first batch a script, a data structure with natural language processing applications. They They call the second batch a story, and they call the third batch questions. The symbols you give back, they call answers to the questions. The set of rules in English, they call the program. You know yourself know none of this. Nevertheless, you get so good at following the instructions that from the point of view of someone outside the room, your responses are absolutely indistinguishable from those of Chinese speakers. Oh, this is that gag from Family Guy where he's in the truck with the one guy who's like, I don't actually speak English, I just memorize the answers to questions, including this statement. And Peter says, really? And the guy says, okay. It resolves this problem by ignoring it. By not caring about it. It resolves this problem by beautifully not giving a single shit about it. It decides that this problem is not interesting and ignores it. And is not relevant to the story it's trying to tell. Then again, maybe I'm just memorizing English. Maybe I've just memorized every possible English statement that I would need in a given day and don't actually speak English at all. Savez-vous? Alors, until next time, instead of reading this article, I'm going to go wash dishes, which is going to be infinitely more pleasant. So... bye.